All right, so our next presenter will be Dr. Gounder speaking about EHE clinical trials. And as I was mentioning, years ago, there weren't any, and now we have some, and more probably coming up in the future, which is just a complete change in the landscape of future options for EHE treatment. All right, your presentation is looking great. I will need your live video and audio. There you are.
Hello?
Excellent.
Can you hear me all right?
Perfect.
Okay, terrific. Well, thank you so much for inviting me. This is my first EHE conference meeting. It's been really, I've learned a lot just in these last 2 hours. So I'm really excited to learn more tomorrow, as well as from the patient experience as well. So my name is Mrinal Gounder. I'm a medical oncologist. I do two things. I'm mainly a sarcoma medical oncologist, but I also wear a second hat in phase 1 early drug development clinical trials across all solid tumors. So I was really excited when we started this trial of a TEAD inhibitor from Vivace Therapeutics a little while ago as part of our larger phase 1 clinical trials group. This study is also being evaluated in mesothelioma and many other types of cancers, but obviously my main focus and interest here is in EHE patients. I also ran the IKINA TEAD inhibitor study that has now been subsequently closed. I will not be discussing that, but what I wanted to discuss with all the participants here today is just give sort of a brief update on what's happening with the TEAD inhibitor. This clinical trial is ongoing right now. There's also many other TEAD inhibitors in development. And today I'll just focus on the Vivace Therapeutics program. So with that said, okay. All right, so I think one key thing that you've all heard again and again is that EHE basically has a fusion of two different genes that normally should not be next to each other. But the consequence of having EHE is that, or these tumors, when these two genes come together, and this could be the CAMTA1 gene or the YAP TFE3 gene. One of the things I want to show you here is the number of mutations you see in EHE. That's here on the left side. As you can see here, EHE, the number of mutations at the DNA level that you see in EHE versus those of other cancers, so other sarcomas particularly. So everything that is shaded here represents a genetic mutation in any one of these genes here. This is a very abbreviated version of all the genes here. So I think the takeaway here is that many other types of sarcomas have lots of different genetic mutations, but EHE is really quite silent, and if there are mutations, they have also been described by several different groups. One from Ohio State, as you heard before, and also from Dr. Rosenbaum here at Sloan Kettering as well, that these could be secondary mutations in other genes such as CDKN2A and such. But today, for the purpose, is really just to highlight that we think that the fusion or the drivers of EHE is really these two fusion genes. And as you can see in this cartoon here, we heard about this LATS1-2 is basically YAP1 or TAZ. Both of them can go into the nucleus of this of EHE and then turn on several number of genes. YAP and TAZ by themselves are unable to function to cause cancer except by going through TEAD. And that's why we are going to be talking about TEAD inhibitors today. In order for this YAP or TAZ in EHE to work, it has to talk via TEAD. And the goal here is how do we basically disrupt this communication between these YAP-TAZ protein as well as the TEAD proteins from making these tumor cells continue to grow. That's the essence of this. It turns out that when you zoom in to this YAP or TAZ and TEAD, that there are many different pockets. So basically the YAP-TAZ fits into TEAD. When it fits into TEAD, that's how this TEAD protein gets turned on. And once the TEAD protein gets turned on, it goes on to turn on many different genes. Many of these genes then subsequently control the growth of EHE. And what you're looking at here, right in the center here, is sort of a 3D crystal structure. And in this pocket, you're looking at the TEAD. There is this molecule or this amino acid known as cysteine. And this cysteine needs to have a process known as polymethylation in order for it to continue its activity. And this, in this purple, what you're seeing is this drug from Vivace Therapeutics, which is VT3989. And basically what this small molecule VT3989 does is it inhibits YAPTAZ from communicating with TEAD, and therefore TEAD is not able to do its downstream activation of tumor-causing genes and proteins. So how is this study set up? So first, as I mentioned before, this is an early phase clinical trial, which means this drug is only available as part of a clinical trial. And the purpose of and it's really part of a early phase 1 and a phase 2 clinical trials. And let me explain what a phase 1 clinical trial is and then a phase 2 clinical trial. A phase 1 clinical trial is typically, its main goal is to take a molecule that seems promising and to administer this in patients with a certain type of disease. This could be hypertension, diabetes, cancer, any number of things. But the general principle is when you take these molecules from the laboratory, you need to, when it enters into patients, it has to start at a low dose. Our goal is not to cause major side effects in patients because this may be the very first time a drug is being evaluated in any human being. So the goal is to start at a low dose, see how patients do, and if the first 1 or 2 or 3 patients do well, then to increase the dose, and then keep going up till at some point you come across a side effects which in general the investigators and the company thinks this is a little too much. Maybe we need to pull back a little bit. And that's the main purpose of that phase 1 clinical trial, is to look for safety, is to see how is this drug being metabolized by the body, and 3, is there any kind of activity that we can see even in this dose escalation part. Okay, so, and sometimes there's also in phase 1, we look at drugs that are given continuously, and we also interrogate different schedules. Maybe the drug needs to be given 1 week off and 1 week off. So all kinds of different sort of programs are put into place, different trial designs are put into place. But at some point, when we figure out the right dose and the right schedule, that's when there is something known as a dose expansion. At dose expansion, typically a few different types of cancers are selected to get more number of patients for that dose expansion to see is this drug really working or not working. So the second part of this Vivace Therapeutics study is here on the right-hand side called expansion cohort. So here there is a drug, a disease called mesothelioma. There is a pleural-based mesothelioma and a non-pleural-based mesothelioma. So there's 2 separate cohorts of patients for which there are this trial is designed just to look at the activity in mesothelioma. There's also this dark blue one for patients with solid tumors, any kind of solid tumors, where this YAP or TAZ gene arrangement is seen, and this particularly lends itself as a nice cohort for patients with EHE. So right now, this study has completed part 1, and it has now moved on to part 2. In other words, the safety as well as the schedule has been mostly figured out, and now the question is, how active is this particular drug in EHE? And just to mention, this is an oral pill that is taken 2 weeks on and 2 weeks off as part of this study. Who are the patients who are eligible? And here's a brief inclusion criteria. This is not extensive, but this is really anyone greater than the age of 18. For cohort 4, which is particularly the one that I just mentioned before, for which EHE is relevant, we need to have documentation, or at least know that the patients have EHE. That means that diagnosis is mandatory. and ideally also need some tissue to confirm that it is indeed EHE and has YAP or TAZ gene rearrangements. In some patients for whom they don't have a tumor tissue already stored as part of a prior surgery or biopsy, they may be asked to do a second biopsy in case there is no prior existing tumor still left behind. What are the other inclusion criteria? One of the inclusion criteria is that the kidney function has to be good. So most people should be okay with EHE, but there are many people who have significant diabetes or uncontrolled hypertension who may have some sort of a kidney damage. So if the kidney is already damaged, then this study drug may not be very effective, and I'll explain to you within a minute why that's the case. Other sort of criteria to enter into the study is that you need to have a normal white count, blood albumin needs to be greater than 2.5, good number of platelets, and then the liver function also needs to be within normal range. Lastly, and this is quite important, is that we must be able to measure disease. If, for example, someone's already had surgery and there is no EHE or some other cancer visible, then they would not be eligible for this study. And when we say measurable disease, it's generally either a CT scan or an MRI scan where the tumor itself on these scans is at least 1 centimeter or more, and it should not have been previously radiated. So there's a lot of nuances and criteria. I'm not going into every single thing, but in general, there must be some active disease that can be measurable to ask the question, is this tumor benefiting from this therapy or not? So let me quickly sort of go over the side effect profile of what has been seen so far. So this is a total of 130 patients. These are not all EHE patients. Most of them are not EHE patients. Most of them are patients with mesothelioma and many, many other solid tumors. In general, one of the biggest side effects that is observed with this drug, this TEAD inhibitor, is proteinuria. Proteinuria is protein that is found in the urine. So for normal, if you have a normal functioning kidney, you should not have any protein in the urine at all. But for some reason, and there are some very good biological studies that have already figured out why protein is being lost through the urine. It's known that these TEAD inhibitors as a class, not just the Vivace, but many of these TEAD inhibitors do as a class of molecule do cause leaking of protein into the urine. You can also have swelling or peripheral edema as a result of this drug as well. But the key thing here is what's known as grade 3 and 4. These are really severe side effects. And what you can see here is that we're not seeing serious or significant proteinuria or even very serious or significant fluid retention in the body. And there is, of course, some fatigue, nausea, and others that you can see listed here. But these are, I really want to sort of highlight some of the issues seen here. The other thing, at least to date, based on these patients, it seems that a lot of these side effects are reversible once the drug is stopped or even when the concentration of the drug is decreased or there is a dose reduction of these things. So again, this is evolving data, and this is just 130 patients, so it's a very, very small number of patients in order to say with confidence how things are. Next question is, are patients with EHE benefiting? And I want to caveat this by saying that this is very, very early in terms of efficacy. In other words, the number of patients on this or any other study with EHE are very few. So you have to take this with a grain of salt. What I'm going to present is you very early data. So here you can see a liver. This is on a CT scan, and you can see circled here is red is an active EHE. Here's another one over here, and is a third one, all part of the liver, but just different layers or sections of the liver. This is before treatment has begun. And what you see here is after treatment, that there is a significant reduction in the size of this tumor from there to here, as well as this one also. There is a subtle decrease that you can see with this tumor. And here also there is a significant decrease after starting this drug. In fact, this patient had what's known as a partial response, which is when you add up all of these, there is a greater than minus 30% shrinkage in the tumor. This is a patient with a partial response of one of the EHE patients who's on study. And this is further quantitated here. As you can see here is when you add up all these different liver lesions, it comes to about 5.5 centimeters. And at the very last follow-up, it's now down to 3.5 centimeters, which represents a minus 36% shrinkage for this particular patient. Here's another patient who's basically before treatment. You can see here the liver. This liver is modeled with several different EHE here circled over here. This is at baseline before treatment, these both of them. And as you can see, after the very first cycle of the drug, you can start seeing some subtle changes that are heading the right direction, and the tumor is starting to shrink again, you can see here. So again, very early changes. Lastly, for people who are interested in exploring this study, these are the sites that it's open in. You can go to a website called clinicaltrials.gov and put in this Vivace drug, and you'll see all the sites that are closest to you. It's open in the United States. It's also open in Australia. So I'm going to stop with that, and I'm happy to take any questions that anyone may have regarding this. Thank you.
Thank you so much. And so one question, and if you can answer this, in terms of the EHE patients who are in the trial, do you have representatives of both types of fusions in the trial?
We've not, it's, like I mentioned, it's pretty early, but I just in my own anecdotes is that most of them have been the dominant CAMTA1. I have not yet enrolled any patient with a YAP.
And also welcoming Denise to the Q&A here. Denise, any questions from you right away?
No, thank you, Dr. Gounder. That was really great overview, breaking down the trial and certainly encouraging information. And I'm glad that you prefaced, with a grain of salt, because it is certainly early days. But it's encouraging that we have a trial that EHE patients can potentially get onto. So, and if some of those patients can benefit from that now so that we can learn something and then expand those numbers, that's really great. I wanted to ask you just in simple, language, if somebody says, they live anywhere in the United States and they're interested, they think that they could benefit from trying this, or they want to try. If someone calls Sloan Kettering, for instance, or any of the sites, what does that look like?
I can't speak about any other sites, but I would say that if they want to, I think the best thing is to call and say, I have EHE. I'd like to be evaluated for this clinical trial. It begins with something as simple as that. And at that point, people need to come to an institution to be evaluated. We generally don't do telemedicine, especially for the very first time. So people come in, they usually have their pathology and other things reviewed here. And during the first meeting is the first time you actually, we evaluate everything, and then also whether the trial is appropriate for them or not. As I mentioned, there is 30 or 40 different criteria to enter into the trial. If it looks like, they may be eligible, it usually requires a second visit or even a third visit to come back, and then to formally screen, to do the testing, to make sure all of this meets. So, and by the time someone says, they'd like to be seen, to the time that the drug gets started on a clinical trial is generally about 3 to 4 weeks, depending on if you're able to get everything lined up and ready to go. I also want to mention that the clinical trial itself does provide funding support for travel, lodging, as well as food and other expenses. For all clinical trials in the United States, as long as if people have insurance, most things are considered as standard of care, and it is billed to the insurance or to the patient. there are some things that are not paid for by insurance, which will be paid for the drug company. For example, the cost of the drug itself. Any research studies that's done, say you're testing a urine for like some biomarker or something that's part of the study, all those things are paid for by the drug company. Generally, there is a pretty decent amount of funding for travel and other things. But most other things like a CT scan, a doctor's visit, routine blood work, EKG, echocardiogram, scans, those are all generally billed to the insurance. So for most patients, whether you are, say, on a regular chemotherapy or you are on a clinical trial, for most part, insurance companies have been pretty okay about paying for these studies. So yeah.
And we did have
Excellent.
an audience question come in here. Are patients with metastatic EHE eligible for the trial?
Yes, yes, absolutely. Absolutely.
Excellent. That hits all of the questions, I believe. Yes. Thank you so much for your time. It's super interesting to learn about these
Thank you.
trials and to, hear these, I know very, very early, but, promising
Looks
results.
promising. Yes, indeed. Thank you.
Thank you, Dr. Gounder.